So I want to start out on the issue of the basic theory of quantitative genetics. For many of you, this will be a review. And I think, nevertheless, I hope you'll be tolerant of it. I have an hour's worth of material and half an hour to give it. We'll see how far I get. What I wanted to do was start out by just saying where the theory came from. And these are some of the major figures involved in quantitative genetics theory. One of them, these here are Francis Galton, a figure of the late 1800s. He died around 1910. He was a younger cousin of Charles Darwin. And his disciple, Carl Pearson, who were both in London. And they put forward statistical laws that Galton's most famous law was called the law of ancestral heredity, which was a statistical summary of the influence of the traits of parents on those of the offspring. It's pre-Mendelian. It does not have any Mendelian underpinnings. It's basically concerned with regression of offspring on parents. The law changed and mutated as time went on. And when Mendelism was rediscovered in 1900, when Mendelian genetics was rediscovered, there was a crisis among these people who have come to be called the biometricians. Galton and Pearson and their students weren't prepared really to deal with Mendelian genetics, and there came to be a controversy with Carl Pearson on one side and William Bateson, the most eminent geneticist, Mendelian geneticist, on the other. And that raged in the 1900s. And it was really resolved, although it took a little while for people to realize it, by the work of R.A. Fisher in 1918. And Fisher, who is also, of course, the greatest mathematical statistician ever, wrote a paper in 1918 which was a tour de force in the sense that it made the predictions of what the covariances among relatives would be under Mendelian genetics, and it gave you machinery for fitting data on covariances and correlations with using Mendelian assumptions and showing you what parameters were involved. I'll be going into this in detail, although not, of course, into the totality of Fisher's extremely mathematically obscure presentation. I've had this guy up here. He's less well known among people who are not animal or plant breeders. Among animal and plant breeders, he's very well known. And that's Jay Lush of Iowa State University, who from the 1920s onward took this quantitative genetic theory that had been developed by R.A. Fisher and also by Sewell Wright and J.B.S. Haldane, who were the great founders of theoretical population genetics, and applied it to animal breeding. It came to be applied to plant breeding too. Worked on many phenomena, involved things like family selection, multi-trait selection, and so on. Published many papers on that. Trained large numbers of people, and Iowa State became one of the great centers of quantitative genetics. And he wrote a book in 1937 called Animal Breeding Plants, which became the classic presentation of this, making it accessible to animal and plant breeders, and as a result of that, it became widely used. So that's a little quick mention of the history. What I want to do is start out by presenting a simple quantitative genetic model of the kind that Fisher and company put forward. And basically what one assumes is that there's a trait. So for the moment we're talking about a single trait. All of this can be generalized to multiple traits. And the scalar quantities can become vectors. And some of the variances become covariance matrices. But this is the single trait version. And you basically say, well, we're going to model that trait as being a sum of pieces contributed by the different genes involved in the trait. You start from an arbitrary starting point, which is often taken, I don't know what the number 4 is doing there, which is often taken to be the mean of the population, but it doesn't have to be. And then you assume that the effects of the different loci are additive. Now that's a strong assumption because it really leaves out epistasis, interaction among loci. But nevertheless, let's make that assumption. So what you're going to assume is that based on the genotypes at this locus, I've shown 2 alleles. The theory generalizes naturally to multiple alleles, in which case these tables get much larger. And I'm showing you that a big A at the A locus will subtract 2 from the value from the starting point. Heterozygote add nothing, and the little a add 3. And there's a similar table for each of these loci. So depending upon the genotype that you have, we colored purple, this genotype, you're going to end up adding up the correct numbers here. And then at the end, after the genotype has done its work and created the genotypic value, you're going to add on an environmental effect. The strong assumption that's made there is that the environmental effect is drawn from a distribution. We've shown a normal distribution here, independently of what the genes are doing. So there's no genotype environment covariances in this version of the quantitative genetics model. So you can see here is a genotype, an imaginary karyotype with different genes that are affecting this character. And for this scheme, we can go down these tables here and figure out what the numbers are that add up. So here's a heterozygote at the E locus. Over here you see that's going to add 0.3. Big A will subtract 2. Heterozygote at the C locus, plus 6. Homozygote at the D locus, little d, 0.7. And finally, heterozygote at the B locus. So you see we've selected contributions from each of the genes here. Then we're going to add on an environmental effect, which just happens. In this case, we made a draw from this environmental distribution. It happens to be 0.9. So we're getting a total phenotype here that is of 10. It needn't be a nice round number, but it happens to be in that case. And if you happen to know this scheme, which we generally do not, we generally don't know how many loci, we don't know what their effects are, what the effects of the different genotypes are, but we imagine that that a scheme like this underlies all the inferences that we're making. And we could imagine that if we knew the genotypes at the relative genes, we can go through, and I've got everything colored in properly here, and assign phenotypes to each of those. So we're imagining that underlying the characters that we're working on is such a model of additive contributions from different loci. Now, we are allowing for dominance, but we're not allowing for either epistasis or covariation between the genetic effects and the environmental effects. Okay, I'm sorry, yes. I went ahead too fast. Here we go. Another model that you will see at the very end of the course, on the last day, I'll talk about threshold models. We won't hear much about them otherwise. You can imagine that instead of the trait being a continuous variable that can be measured, that instead it's an all or nothing trait. The classic example that Sewell Wright did when he introduced this model was hind limbs of guinea pigs, whether the guinea pigs had 3 toes on the hind limb or 4 toes on the hind limb. And you can code those as 0 and 1, and you can imagine that there is a threshold. So if we have a threshold of 9, we can go down these and say, well, underlying is a quantitative character that we're not seeing, but what we actually see is then determined either to be 1 or 0, depending on which side of the threshold those values are on. And we'll see some machinery involved with threshold models at the very end of the course. Those are it's evolutionary, not quite quantitative genetics. It's evolutionary discrete genetics, but with an underlying quantitative genetic model. To show you how the genes create distributions of these traits, I wanted to use a few examples. Again, I'm emphasizing that what I'm showing represents a level of knowledge that you don't actually have. You don't know what genes are involved. But we'll see in a moment that quantitative genetics gives you rules to connect observables with each other. And that marvelous fact comes from R.A. Fisher's 1918 paper. If you had a single locus with 3 genotypes, and they happen to have genotypic means of 2, 4, and 5, imagine that the effect of the environment, so there's only one locus involved in this trait. Imagine that the effect of the environment was not I'm violating this model I showed you in a moment ago. I'm saying that the amount of variation caused by the environment is different in the different genotypes, because what I'm going to assume is that on this scale, the environmental effect adds on a log scale. So these are little log normal distributions. They're slightly different widths. I just wanted to do that to get a little more realism into the model. If you have a small amount of environmental variation, of this log normal environmental variation, you would see a distribution like this. If you were working on an organism and measuring this trait, you would notice that there were 3 groups of values, and you would start thinking about doing Mendelian genetics on it. So in this top figure, you're in the realm of Mendelian genetics. As you add more environmental variation, you start getting a situation like this. This distribution is fattened out. These two have fattened out to the point of merging with each other. Now, it's not quite so clear that you can make a clean Mendelian analysis of this, but you probably could. With a little more environmental variation, all 3 distributions merge with each other. At that point, you can't see the genotypes. You cannot neatly classify individuals into genotypes. And at that point, you're going to have to use quantitative genetics. So there you see the sort of boundary between a Mendelian genetic analysis and a quantitative genetic analysis. Likewise, if you have many loci, suppose I had one locus and environmental variation around it, and there could be enough environmental variation that these 2 distributions start to merge. What I'm going to do here is show you the effect of adding not more and more environmental variation, which is what I did in the last slide, but having more and more loci. And I've scaled these so that the total variance of the trait is the same in all 4 of these cases. And but I've added more and more loci, and I've scaled them so that the environmental variance is 30% of the total variance. If we have one locus, it looks like this, and you can see 2 peaks. So you have a hint of the Mendelian genetics. One can make a rather noisy Mendelian genetic analysis from that. If you have 2 loci, there are 3 peaks, but they're starting to merge here. It's getting hard to tell them apart. If you have 5 loci, now they're all merged together. You cannot really see the effects of the individual loci. It's beginning to look like a normal distribution. Many of you will be familiar with the fact that if you have distributions of sums of random variables, as you get more and more variables contributing to the sum, each contributing a smaller and smaller amount, you approach, the distribution will approach a normal distribution. Here you see it very nicely. This is actually a mixture of peaks from 10 loci. But you really can't tell. If somebody came along and said, I'm going to give you a sample from this distribution, tell me whether that's a normal distribution, you'd have a very, very hard time detecting that it is not a normal distribution. It isn't the normal distribution, but it looks to all the world like a normal distribution. You'd need huge, huge sample sizes in order to detect the departure from normality. So very quickly, having multiple loci gets you out of the Mendelian realm and gets you into a realm where distributions magically become normal, even though originally they weren't. Okay, and here is a case I won't go through in great detail of two loci interacting. These are the genotypic means. If you work your way through, you'll see that they're not fully additive. There is some epistasis here. For example, let me see. Yeah, this, if you go up this column, the heterozygote at the A locus is intermediate between the homozygotes. If you're in this column with little b, that's not true. You'll see little interactions there. If you take this scheme and add more environmental variance to it, it's a two-locus character. You see at first you're in the Mendelian realm, and then quickly you actually go through a stage where it looks like there's only one locus contributing, but that's not true. And then finally you arrive at a distribution. Here I'm mixing in log normal environmental variance. So it's once again somewhat asymmetrical. Okay, so those are some examples of the approach to the realm in which you just got a distribution of the character, usually something like a normal distribution, and you want to do some kind of quantitative genetic analysis. And here we get to the magic Fisher machinery, and it's a rather hard sell, because you start out with, you say, well, suppose you knew what an individual locus was doing, and we define some quantities, some of which appear to be somewhat imaginary. And then later I'm going to show that those yield up observables that can be related to other observables. The things we're seeing here aren't observable. Here's an underlying locus. It's got two alleles, A1 and A2. The dosage of A1 is given down here. So this is the A1 homozygote. This is the A2 homozygote. Suppose the genotypic mean contributions to this sum of values coming from all the loci were given by these dots here. So the locus, the A1 locus, is almost dominant. The heterozygote is almost as high as the homozygote. What R.A. Fisher said to do is something that doesn't strike you as an immediately obvious thing to do, and that was to fit a straight line through this data and define some quantities that are going to be called additive effects and dominance deviations. The fit is a least squares fit where the points are weighted by their frequencies in the population. Now remember, this is all something that cannot be done in practice. You won't ever be asked to do this because you won't ever know exactly what contributions this particular locus is making. Maybe with modern genomics one can start getting there. But you imagine yourself doing this, and you weight these. If the A1 and A2 alleles are segregating in a natural population, or in your natural or artificial population, in Hardy-Weinberg proportions, you'll have frequencies of these three dots of p squared, 2pq, and q squared. You do a regression, weighting a least squares regression, weighting so that what you're minimizing is the square of this departure from the line times the frequency of that genotype, similarly at the other two. Having gotten that, we can define additive effects of the A1 and A2 alleles. And what Fisher said is, well, this departure from the starting point, which might be the population mean, is the sum of two additive effects that are called alpha 1 of the A1 allele. This one is this departure here, that is from the population mean to the line, is two effects alpha 2. In this case, they're negative. Those define alpha 1 and alpha 2, and by the linearity of things, this one here will be a sum of 1 alpha 1 and 1 alpha 2. So we could, in principle, if we were omniscient and we knew everything, we could, in principle, calculate the values of alpha 1 and alpha 2. The actual genotypic mean contributions from each genotype are not on the line, but they're off the line, and the deviations, the further deviation that you have to go from the line to the genotype defines three quantities, delta 1, 1, delta 1, 2, and delta 2, 2, that are called the dominance deviations. Well, why are we doing that? We're assuming, I should say, Before I tell you why we're doing it, the assumptions that I've mentioned already are you're in Hardy-Weinberg proportion, so you're implicitly assuming random mating. We're going to be considering the contributions coming independently from each locus, and that implies that you have linkage equilibrium between all loci. You have no association between presence and absence of genes at different loci. The environmental effects, as I mentioned, are considered to be independent of the genotype. And we're also going to be assuming that the environmental effects, at least in simple naive cases, the environmental effects, the environmental contribution is independent from individual to individual. There's no substructuring of the population. Quantitative geneticists working with animal and plant breeding have to worry about whether that is true. For example, it's well known in animal breeding you have to worry about herd effects. Maybe one farm is feeding their cows better than another. And so individuals on the same farm have correlated phenotypes, correlated environmental effects from that. And a lot of effort goes into correcting for those. And we've assumed away epistasis. There's no interaction between loci. So as a result of all those assumptions, you put together all of those assumptions, and what you've got is that if you're looking at an individual and considering its phenotype as a sum of contributions from each locus, but also at each locus above 2 alphas and a delta, the 2 alphas that you'll find at a locus are independent of each other. They're independent of each other because which allele you get at each of those 2 positions is independent by Hardy-Weinberg. So that's built in. Now here, as a result of a lot of algebra involving the least squares fit, it turns out that the alphas and delta in a single locus are uncorrelated. They're technically not independent, but they are uncorrelated. That's all we need. As a result of doing a lot of algebra involved in the weighted least squares. So I've just said, trust me. Otherwise, we would spend half an hour on that one. And of course, the alphas and deltas contributed to a phenotype at different loci are independent by linkage equilibrium. The genotypes at different loci are not in any way associated. And the environmental effect is also independent. So the resulting phenotype is then, you can consider now as a sum, not just of pieces from each locus, but of the alphas and deltas at each locus and the environmental effect. Okay. Well, you may wonder, well, where has that gotten us? We've divided up these unknown genotypic effects into unknown smaller pieces, but so what? How is that helping us? It's just getting us further from reality. Well, what we can do is imagine rearranging that sum to put all the alphas together and all the deltas together. Now you can see that the phenotype is the sum of 4 parts: the starting value, or possibly population mean, the total additive effect, total of all the additive effects, the total of all the dominance deviations, and the environmental effect. So you can think of it as mu plus A plus D plus E. All of these are uncorrelated with each other. It follows that the variance of P, if we look at the distribution in the population, the variance from individual to individual is a variance of this sum of uncorrelated pieces. It follows from that it's the sum of the variances of all these individual pieces. And we can take the total variance contributed by the alphas and call that V sub A. The total variance contributed by the deltas, V sub D, and the variance contributed by epsilon, V sub E. There's no variance contributed by mu because it's a constant that's the same with everybody. So now what we've done is break up the variance into 3 parts. But again, so what? And each of those parts consists of contributions from many loci, and you don't know how many loci, and you don't know what exactly are their genotypic effects. Well, the rationale for why you or R.A. Fisher are doing this will begin to become clear as we consider this slide. We're going to consider the covariance between two individuals that are relative. So we might have a parent. The one we'll be most interested in is a parent and an offspring. But there'll be things like covariances between siblings, covariances between nephew and aunt, for example. Each of those phenotypes is a sum of uncorrelated parts. And so it's a covariation of one sum with another. Now, it happens that just by doing trivial algebra here, that when you have random variables x, y, and z, and they're uncorrelated with each other, I've said independent here, but you only need uncorrelated, that the covariance of this sum with that sum is the variance of the part that they share, if all the pieces are uncorrelated with each other, which they are in our model. So if we take the phenotype of individual x, and it is a sum of these alphas and deltas, I've left off all the super and subscripts because to make it clearer. And there's another individual y. This might be a parent, and this might be its offspring. And we're imagining we have many of these parent-offspring pairs. And we want to look across those parent-offspring pairs and ask, well, with that set of data, what's the covariance between the parents and the offspring? The parents are a sum of pieces. The offspring is a sum of pieces. The Mendelian genetics guarantees that some of the genes in the parent show up in the offspring identical by descent. I'm not going to go into a long discussion of identity by descent, but simply that they are there because of Mendelian genetics. So, for example, at the first locus, one of these two alphas here is a copy of one of these alphas up here. By Mendelian genetics, a random one of the two. So what you can show is that half, if it's a parent-offspring pair, half of the alphas in this sum are identical as a result of Mendelian genetics, and they must be making the same contribution to the phenotype. And so we can calculate, based on the degree of relationship that we're looking at, that F1 here is 1/2 for parent-offspring pairs. And you can easily do it for other relationships. Similarly, the deltas will be shared when both of the genes in in one individual are identical by descent to both genes in the other. Now, in parent-offspring pairs, that won't happen because one of the genes is identical. The other one just was drawn from the population at random. But if these were, for example, two siblings, then what you can show is that 1/4 of the time, the deltas they contribute will be identical because you'll have exactly the same genotype at that locus as you do in the other sibling because of the descent of those genes from the immediate parents. So for any given relationship, you can calculate what F1 is and what F2 is. And our assumptions that we've made also guarantee that the environmental contributions are not correlated. Now, in real life, of course, they are, and you have to worry a lot about that. For the naive presentation of the theory, they're not. So the results you get out, this is an R.A. Fisher result from his great 1918 paper. Fisher, by the way, was nearly blind, which saved his life in the First World War. He wasn't drafted. But because he was nearly blind, he had very poor eyesight, he did all his algebra in his head. Because of that, he assumed everyone else could too. And the result is, of course, the 1918 paper is an impossible read. Whole seminars of theoreticians have sat down and spent an entire quarter trying to get through the paper. But one of the central results he gets is that the covariance of offspring of a given degree of relationship then turns out to be this fraction F1 times the additive variance. If you share half of the alphas, then the shared part of those sums of random variables would be the variance. So it will be the variance of half of these alphas, of the sum of half of these alphas. And that turns out to be half of the additive genetic variance of the trait for the dominance deviation. F2 of it, which in the case of siblings is 1/4, in the case of parents and offspring is 0. So there's a useful formula. Now you can begin to see why in R.A. Fisher's theory, you start with a whole bunch of loci that you don't know. You don't know they're there. I can measure the length of my thumb. How many loci contribute to that? I don't know. It's large. But now you can say, aha, we have a formula for something we can measure, the covariance of relatives, in terms of these mysterious quantities at V sub A, these variance components, V sub A and V sub D, with coefficients that we know from Mendelian genetics. So now we can say that we can use the covariances that we measure to estimate VA and VD. We can accomplish the variance breakdown without knowing which genes are involved, how large their effects are. And this will work, by the way, for even if the trait is controlled by only one locus with some environmental variation. That works perfectly. If it's controlled by 1,000 loci, it works perfectly. So what it does is it gets you away from having to know the genetic basis of the trait. And that's why it's been so useful in animal and plant breeding over the last century, because we didn't have the genetic information, but they could relate these observable variance components to each other. You may say, well, this breaking down of variances into variance components looks familiar. Doesn't it have anything to do with analysis of variance? Well, if you know analysis of variance, ANOVA, you may know who developed it. It was R.A. Fisher. And when did he develop it? A couple of years after he did the variance breakdown for quantitative genetics. So it was all part of the same intellectual machinery as analysis of variance. In effect, each additive effect is a single factor effect. In the variance components, it would be like a row effect or a column effect. The dominance deviations are two-way interaction effects between the rows that, the directions that correspond to the two copies at a single gene. And you've forced a lot of the higher order interaction of variance components to 0 by the assumptions that we've made. And the least squares regression that's used is in the analysis of variance, you're implicitly using something like that least squares regression. So it's really the same machinery. And I've said all this. You can work out the F1 and F2 from Mendelian genetics, and you get this formula. So the important case that we're going to use is parents and offspring. And there, a fraction 1/2 of the alphas is shared and 0 of the deltas. Two individuals might contain the same genotype, but if it's not because of the genetic relationship, but simply accidentally, that doesn't bring a term in for V sub D. Okay, so the covariance between parents and offspring is half the additive genetic variance, half the portion of the total variance that comes from the additive terms. Now there's a quantity you will have heard of called the heritability. That simply is the fraction of all the variance in the phenotype that comes from this additive term. It's important to remember that it's not the fraction of genetic variance, it's the fraction of additive genetic variance. It's part of the genetic variance. It does depend on the gene frequencies because remember, we established those alphas by a linear regression where we weighted by the gene frequencies. That means that in two different populations where a given locus has different gene frequencies, the alphas will be different. And the result will be the contributions to the V sub A term will be different. And so these quantities are not magic numbers that automatically associate with the trait. They're specific to the particular population because they depend on the gene frequencies. And as you go from generation to generation, as a result of genetic drift or natural selection or other evolutionary forces, those gene frequencies can change, and thus the variance components can change. And that's, in a way, comforting because you think that if a population goes on and loses all its genetic variation and everybody's got the same genotype, you'd hope the theory would tell you that the additive genetic variance and the dominance genetic variance go to 0, and they do. It's comforting. So they're different in different populations. I should just mention that for historical reasons connected with his machinery of path coefficients, the heritability turns out to be the square of a quantity h. That quantity h, defined by Sewell Wright, is never used by anyone in quantitative genetics. So heritability is always written h squared, and there's never any need to square root it. It's kind of irritating, and you kind of like to square root it, but then what you get isn't useful. So here is a slope. This is I am going to finish up before we get to the end of the half hour. Here is two normally distributed traits that have a certain additive genetic variance. In this case, I believe I have made them have a heritability of, I think in this one it's a heritability actually of 1. Okay, well anyway, there is a certain, I'm embarrassed to say I don't remember when doing this what heritability I actually assumed. Here are the parent and offspring pairs of a distribution in the particular case that I simulated from. This is done by simulation. There is the true relationship. It is close to the fit to these 200 points, but it isn't exactly the same. The true relationship between the genotypic value of the parent and the genotypic value of its offspring. So that's like a father-offspring pair, for example. It's not taking both parents. It's taking one parent. Now, if you have that, this might be what your data might look like. The expected slope between the parents and offspring is half the heritability. If you take a parent and you breed from them, you say you go out into this population, out into this herd, you take all individuals, you find some individuals who have this as their phenotype, and you use those, mate them with random individuals, and produce offspring. What this says is the offspring will be distributed in this slice. Now, this is a bivariate normal distribution. In a bivariate normal distribution, the conditional distribution of a slice like this is a normal distribution whose mean sits right on the regression line, and the theoretical regression line in this case. So the mean of their offspring, if you do enough of them, will be here. So that says that selecting from parents who are here, you get offspring which on average are there. 10 is, by the way, the mean of the population in both the parents and the offspring. Excuse me. And from that, you can get that if you say to yourself, okay, we're going to do an artificial selection experiment. We're going to take our herd and we're going to breed only from individuals that are above this cutoff, which is somewhere near, it's a little bit below 11. We will breed only from them. Well, we've seen that for each slice here, the mean is given by bouncing off the regression line. It follows by the linearity of this regression line that the average response is the average of the parents and taking the average value of the parents, the mean of the selected individuals, and bouncing off the regression line. So the gain that you get selecting on one parent, breeding from, say, fathers who are out here and mating them to random individuals, is equal to what you would get from this. And the result that you get is this. Selecting on one parent, it would be half. The response you get will be half the heritability times something called the selection differential. And whoops, I'm sorry. The selection differential is how far above the population mean the average parent is, the average parent that you actually breed from. That's called the selection differential. And if you do the selection not on one parent but on two parents, the selection gain doubles, approximately, to good approximation. And then you get what's called the famous result, that's what's called the breeder's equation. It simply says the response to artificial selection is equal to the heritability times the selection differential. So by estimating variance components, you can infer the heritability, and that gives you a prediction of how much gain you're going to get from artificial selection. And you can see immediately that this could be very important in animal and plant breeding, and it is. And so I will go in a month from now, and I bet you that Patrick is going there too, to the World Congress of Genetics Applied to Livestock Production. Are you going to be there? In Vancouver. Yeah, so he's going. Where I will find animal and plant breeders, well, animal breeders mostly, who are very practically concerned and who have millions of dollars invested in their selection programs. And the use of this equation and its fancier relatives, its matrix and vector relatives, and all kinds of elaborate breeding designs and all kinds of elaborate schemes for environmental variation is the foundation of a lot of what they're going to do. And this World Congress is not 100 people gathered in a room, but it'll be rather larger than that, and there'll be rather more money at stake. So with that, I wanted to finish the tour of simple quantitative genetic theory. You'll be hearing a lot more about it. Here are some of the references. This is Fisher's paper. If you want to get a massive headache, you can go and read it. Jay Lush's book, it's actually on, a version of it is online, one of his later editions. You can read that. Here are two books on quantitative genetics, two major textbooks, both very good. Douglas Falconer's classic 1960 text, which has been kept updated, and it's gone to 4 editions with Trudy Mackay as the co-author. We can discuss later whether she's Mackay or Mackay. And both is the answer. And Michael Lynch and Bruce Walsh have written a new book on that starting in 1998, Genetics and Analysis of Quantitative Traits. So what I have to say about both of these books, if you seek insight, you will find it in either of these books. Bruce also has chapter drafts of the volume 2, which he is working on with Lynch and Walsh. I have a freely downloadable textbook that's for my theoretical population genetics course. At least one person in the room has taken that course. And that can be downloaded from a website in my lab. Chapter 9 goes through the presentation with somewhat more detail